minute. Okay. Since we have to transmit also to YouTube, I have to set up some things to the transmission. Okay. Okay. So, okay, it's ready. So, hello everybody. Welcome again to the lectures of the International Society for Comparative Psychology. Today we have the honor to have with us Professor Paco Calvo from University of Murcia. So Paco, thank you for being here with us. It's an honor for us to have you here. So if from this moment you can share your screen if you wanted.
Okay,
so we can
good.
Start.
Yeah, would be good to know if it works, right? Okay, let me see. Is it working?
Yeah, it is working.
Great. Okay. Well, yeah, thanks, Eduardo, for the invitation, to all of you for being here. Actually, I wasn't quite sure what sort of focus I would be using for this talk, because there are a number of issues, or I have a number of different headaches as of late, right? Some of my headaches have to do with the very idea of planta sapiens, or of plants being sapient. By this I mean either the talk of plant intelligence or plant cognition, and of plant sentience, right, as such. And a different issue has to do with not with the sapience of plants per se, but with the impatience of we humans as researchers, right? So this is something that I've been struggling with lately, because I have the feeling that we have become increasingly impatient. So I hope, Eduardo, I'm not sure who is in the room, but I hope you managed to gather a handful of grad students and early or young postdocs, because there are a few messages that I would like to be able to discuss with you all, but especially to see if we are able to resonate with some of these messages, right, related to how impatient we have become in our search, in our research, right? And of course, the emphasis will be on what it implies to the very idea of performing comparative studies, right? So one of the things let me push, okay, here on the side, I can see you now. Yeah. So if we are gonna try to understand how the psychology and the behavior of different species converge or can be compared to one another to some extent, one of the risks we run into is the risk of relying on some sort of toy comparisons, like so to speak, to take for granted that the comparison can be established, that there is some sort of metric in which, or on top of which we can lay down the different parameters and the parameter space itself on which to establish the comparisons when it comes to comparing the behavioral repertoire of different species, or when it comes to studying the degree that their psychological states are being shared with one another, right? On top of that, we run the risk again of falling prey of many different sorts of anthropocentrisms, right? So one has to do maybe not just with what we mean by those psychological predicates. Are we speaking of decision making, or attentional processes, or memory storage, or learning, or whatever? But not just that, but what sort of school of psychology are we thinking of? What sort of psychological methodologies or toolkits are we employing, right? So are we trying to perform or to establish some sort of comparison metric that relies on behaviorist posits, or is it a cognitivist approach, or is it other brand or other school of psychology we are thinking of? Okay, so sometimes when we speak of a comparative approach to the study of intelligence or sentience, we are maybe sometimes not careful enough as to what sort of theoretical endorsements or what sort of a priori or implicit theoretical assumptions we are carrying with us, right? So that's something I don't want to get to discuss too much, but I will save a few slides for the end. But first, I think it's also important to think of what we mean by behavior when we are trying to perform these comparisons when species are not that close to each other as we would like to think when it comes to thinking of plant behavior, of course, right? So one of the things we did a few years ago, together with my colleague, with Charles Abramson, was to think, okay, let's see how clear we are on definitional issues, right? If we are speaking about intelligence, about cognition, about behavior, on top of what sort of psychological or school of thought we are thinking of, we need to decide whether we are on the same page as to what we mean by those terms, right? And what we found, and many of you are, of course, familiar with, but this is good to remind ourselves of this issue, is that most of the textbooks that we decided to check didn't contain a very explicit definition or a definition at all of, for example, behavior, right? There was not consistency either in between the disciplines that were trying to tackle the definitional issue. And some authors, like Levitis et al., they found like 20 plus or 25 or something definitions. They alerted, the red flag being raised was that behavioral biologists, they don't even agree on what constitutes behavior, right? Some other authors, now from within the plant science field, at least showed some promise insofar as they thought that the study of plant behavior to some extent could contribute to rethinking the way in which we are approaching the very idea of behavior, or the way we are studying behavior, what we mean by behavioral studies and what we mean by behavior itself, right? So maybe by looking at not animal systems, we might end up thinking, oh, we have a better picture, a clearer understanding, or a wider understanding of what we mean by behavior. Oops, what happened here? Yeah. So just take one example of a definition. Some people would say that by behavior they mean the type of coordinated responses, internally coordinated responses of whole living organisms. That is, we could be speaking of a collectivity, or we could be speaking of an individual agent to external or to external stimuli. And of course, this would exclude the type of responses that we would be thinking of as simply developmental changes. So in the case of plants, this is even more difficult to make sense of, because sometimes when we speak of plant behavior, we put plant behavior in terms of patterns of growth or developmental changes. The borderline is really thin and it's easy to be crossed. And to conflate what might be a genuine pattern of behavior, a behavioral response of the type that we could unearth through time-lapse photography, with mere or developmental changes per se. Very interesting to study, but nothing other than developmental changes, right? So maybe here the borderline is more difficult to draw sometimes in the case of plants. And another issue is that some people might be thinking more in particular of hold on, let me, there you go. I have this. Basis, okay, there you go. Of the type of consequences, observable consequences that we can operationalize, of course, of the choices an organism is making in responding to those stimuli, right? So we could be speaking of choices, and that would give us a good entry route, right? A good entry point to the behavior, the genuine behavior of plants, right? So this is just an example of someone might be thinking of as a behavior, right? A behavioral pattern according to this definition. And this is a cuscuta, it's a parasitic plant. So cuscuta doesn't have any chlorophyll, so it cannot do photosynthesis, and it has got to find a host that it can latch onto and twine around and drill it and suck up its nutrients. So here you have a cuscuta. If you saw it, you have a cuscuta searching for a host. In this case, it's a tomato plant, which is really juicy and able to target it and control the pattern of growth in such a way that it's able to secure its nutrients, which by itself it lacks. It doesn't have the ability to make its own sugars through photosynthesis. Now, that might be at the beginning, someone might think that that's not really an example of a choice as per our definition. But if you think of cuscuta, well, you look at this. This is a good exemplar. So this cuscuta you have here, this tiny cuscuta, this is what it becomes once it has found a host and it has developed into a full-grown and mature cuscuta plant, right? So it's really invasive. It's doing really well. And the choices are clear when you are confronting, or when cuscuta is confronting two different targets. Say this tomato plant as the one you saw in the previous time-lapse footage, as opposed to wheat. So if you are putting those two host trees, potential hosts, in front of a cuscuta exemplar, they will be able to pick, to choose one and not the other, right? In doing so, they will be making a choice based on the amount of nutrient and many other characteristics that makes one of them more appealing than the other, right? So in this case, for example, if you're thinking of the approaching maneuver of the cuscuta, we could be thinking of how they can tell the size and the shape of the potential host, how close or how far away it is, and the chlorophyll content that it has. And it's able to tell all this out of information being provided by light itself. So think that this cuscuta has some light sensitive proteins, in this case cryptochromes and phytochromes, that are sensitive to the blue and the red and the far red parts of the spectrum. So different wavelengths, and they have different proteins sensitive to these parts of the spectrum. And if you think that a plant is absorbing red and blue, and the far red is being reflected or going through, that's why you see the greens, right? It's absorbing the blue and the red. By picking on those light profiles, it's able to, for example, tell the ratio, the amount of red to far red light. So the amount of red to far red is giving some cue to the plant as to the size, the proximity, or the interest in the potential host that is approaching, right? On the other hand, it's not just a matter of exploiting that light-related information. So it's also picking on the type of volatiles, so chemicals being emitted by the host plant. So the tomato plant is emitting these volatile organic compounds, and the plant, the cuscuta, can sense those volatiles. So what it's doing after all, the plant is actually integrating all these informational channels. All these stimuli are being integrated, are being sensed and integrated. So in this case, both the ratio of red to far red light, the blue light, and those volatiles. There is a lot of information yet to be unearthed. So there is a lot of research waiting to be done as to how those informational channels are being connected to one another. So that's part of the puzzle. We still don't know. There could be some involvement as well of sound in the form of phytoacoustics. It's a really recent field of research. But all in all, some of this information, at least light and chemicals, we know for sure they're somehow being integrated in order for the plant to make up its mind, if I may, as to what sort of host is worth, or what sort of direction is worth growing into, right? To pick or to go towards the tomato plant and not the wheat in this case. By the same token, when the cuscuta has already twined around the stem of the host plant, in this case a tomato, it can tell through physical contact and through the chemicals being emitted by the host, together with the information it was already processing in relation to those light regimes, about the nutrient content, the chemistry of the surface, the vascular status of the plant. So all that information is very much taking into, oh, what's happening here? Eduardo?
Give me a minute. Somebody's using like the...
Somebody's painting on the screen?
Yeah. Yeah. Please stop doing that.
I think it's a kit. It must
Yeah.
be video.
Wait a minute.
Yeah.
Capaz que puedes eliminar la función que le permite a la gente cambiar o pegar sobre la pantalla.
Yeah. Just give me a minute, Paco.
Yeah, sure.
Thank you. Can you share again your screen, please?
Hold on. There you go.
Okay. Thank you.
Okay. Clean screen.
Thank you so much.
Okay. So now, if the name of the game here is integration, so yeah, maybe that guest could be muted, because I think it's unmuted. So that kid who is drawing on the screen is also screaming or something. So
I already kick out. So
yeah. Okay. So if the name of the game is integration, we just mention chemicals or we mention light, but here you will see it took us in some other work, we spend some time listing the type of external and internal, both biotic and abiotic signals that plants could be sensitive to, right? So all in all, the idea is that they've got to be integrating all this, incredible long list of signals, and in order to provide a systemic response. So some sort of systemic signaling has got to take place for the plant to develop, to put forward a strategy as to the choices it's making in terms of, for example, in this case of in which direction to grow, which host to choose, and like this, the same for any other competency we might be interested in, right? So now these comparisons, when we speak of systemic signaling, when we speak of integrating information channels, of course, in comparative psychology, we are far too used to thinking, oh, there's got to be some neural hardware doing all this, right? And intriguingly enough, we know those chemicals that when they are being deployed and synthesized by animals, we are happy to understand them for their signaling roles. So in this case, they are neurotransmitters. Like think of dopamine, histamine, or noradrenalines, serotonin, glutamate, GABA, whatever. Any of these signaling molecules, when we know that plants produce them, right? Some of them we know they play a signaling role, but for some reason that really escapes me, and this is amazing to see how the bearing of this, the weight that language carries with it, we are not quite happy with calling them neurotransmitters, even though functionally speaking, the role they play is the same. So it's a signaling molecule, properly speaking, if you think of GABA, for example, right? Now, some of the discussions we've been having with mainstream plant scientists, they respond along the following lines. Oh, this couldn't be a neurotransmitter because there is not a synapsis in the first place, right? So, but in this case, in a comparative approach, functionally speaking, it would be way more enriching to be thinking, well, if there is no synapses, if there are no synapses in the case of plant communication and plant systemic signaling, the integration of signals within the plant to produce this global response, then maybe the conclusion to reach is that, oh, maybe then having a synapsis was not what we were after. Maybe we need to be looking at similarities at a higher, at a more abstract level of description, right? So we have many examples. Think of the way glutamate is increasing the concentration of calcium in the plant, and this is allowing for the plant to assist in the propagation of action potentials. So when a plant is being wounded, think of now you're going to see this plant is going to be wounded by this caterpillar. You'll see at the bottom a caterpillar munching on the plant, and you'll see how there is a spread of activation, how calcium wave spreads all over the plant, carrying this information to distal parts. So to prepare to put the plant ready for this systemic global overall response that this need to be mounted against this herbivore attack. So you see now the caterpillars munching on the plant, and then you see how it's responding systemically, right? So this signaling role, based on the spread of calcium wave by being triggered by glutamate, is what we would be thinking is required for a signaling molecule to play that role, right? So think of here we can induce it with the induction of glutamate on the cat, you see? The glutamate is here. Now you see the spread of activation all over the plant, right? So when we are able to move away from a more animal-centric or a neurocentric understanding of the nuts and bolts of how these global systemic responses are orchestrated, then we can start to understand plant behavior in a whole different way, right? So in fact, we can speak of decision-making, the choices being made by plants or the way they communicate. We can be speaking of kin recognition, how they recognize kin versus non-kin. We can see how plants are sensitive to risks, and depending on the potential risks, they decide to behave, to grow, to move in one direction or another, or put up a whole different set of strategies, of skills, or toolkits. They can behave anticipatorily, so they can decide ahead of time in which direction to grow. If, for example, they are picking on a gradient, and they can sense a gradient of nutrient here, and they might say, hey, maybe despite my roots sensing more amount of nutrient in absolute terms here I can pick that I can sense that the nutrient gradient is on the rise, so it's rising up, and it pays off to grow in this direction because in the future I can anticipate that it will be better off if I will be better off by growing in this pot and not this other pot. So they can behave anticipatorily in this case. They can learn, or we'll see if they can. That's an open question. They can compete, they can mimic, perform numerosity, collective intelligence, if we could use not individuals but collectives as another type of measurement, and a large, etc., right? So when we are doing all this, again, we are not, I don't think we are still quite ready to treat plants or to have or to see plants on the same page, right? So there is some research by some of you may be familiar, by Jonathan Birch, Jablonka, Eva Jablonka, and some others, colleagues, and they've been trying to apply these comparative studies truly across the board, at least within the animal kingdom, right? Within the animal kingdom. So they might say, or in fact they do propose a list or a number of indicators or markers of sentience. For example, in this case, I'm talking sentience, not cognition. And they say, well, look, we cannot observe sentience directly, that's for sure. We can infer, or we can posit the existence of sentience in the case that some form of life, animal form of life, is able to exhibit certain degree of sophisticated behavior. say the amount of integration, how integrated the behavioral pattern becomes as a result of this integration of all these external and internal signals. How, for example, they respond to anesthetics. Think of, for example, how plants or how animals could respond to being anesthetized or behave aversively against some nauseous stimulus or whatever. Whether they can perform some enhanced form of associative learning, okay, they can learn in some form that goes beyond basic associative learning. And for some reason, they have developed a theoretical framework in which we could use some sort of metric space in which any organism whatsoever doesn't need to be a primate, doesn't need to be a mammal, doesn't need to have a neocortex. So this applies to birds, to cephalopods, to invertebrates, to any form of animal life whatsoever. Insofar as they check these boxes, or they are able to integrate, boom. They respond to anesthetics, boom. They are able to exhibit enhanced associative learning, boom. So if they are able to exhibit or to tick or to check all these boxes, then we might or we would have good reasons to believe that to some extent they have some form of sentience, right? So the argument would go. Now, interestingly, one example is integration itself. So we've seen that the vast amount of signal integration taking place in plants. So that's beyond the question. We know integration takes place because they need to provide a response that is adaptive at a global level, right? So plants need to respond adaptively, flexibly, anticipatorily. So in order to do all this, they need to integrate all these informational channels. And that's beyond doubt, right? But how about the rest of it? How about the way plants respond to anesthetics, or whether plants are able to exhibit associative learning? So in the case of anesthetics, it's interesting because we know that plants can be anesthetized with the very same compounds that are being used to put animals to sleep. So in this case, we took a Venus flytrap to the vet school here in Murcia, right? And literally, so we took the Venus flytrap, if you can see it here, we put it here at the vet and use the same molecule, in this case, isoflurane. So the very same of volatile anesthetic that is being used to dose off dogs or cats, you know, your pet. Now, the fact that plants can be anesthetized to some people are really, it could be really shocking, but we know it shouldn't surprise us at all. If you see, we can anesthetize mimosa plants, we can anesthetize Venus flytraps, we can anesthetize pea plants, the one we will be discussing in the case of associative learning, or Drosera, another carnivorous plant. So just look at this plant. This is a case of mimosa. You know that mimosa, we don't even need to time-lapse them because you can see their folding behavior to the naked eye, right? So when you touch mimosa, it will fold its leaves. You can see that, okay, folding in real time. You touch it and it folds. Now, when you anesthetize it with isoflurane, this is what happens. You touch it. In the case, oh, sorry, I should have checked this one first. You see this one? So when you touch it, it just doesn't respond at all, fully flat. Okay? So here, okay, can you see? Yeah, can you see this? So here it's not being anesthetized. When it's being anesthetized, it just doesn't respond at all, okay? And here, when you see the plant, you allow some time for the plant to recover. So when the plant has come out of anesthesia, okay, you can see that the plant is actually responding, okay? You see now how the plant, when it's being touched, it's responding, if only slower, okay? So it's taking a little bit longer for the plant to respond. So in a sense, the plant is coming out of anesthesia. So the anesthetics, the drug is wearing off. So it's like when an animal is coming out of anesthesia, and it takes some time for the animal to become fully aware and fully responsive. In a sense, the very same thing is happening to the plant when it's coming out of anesthesia. So if the sensitivity to anesthetics is one of the indicators or the markers of sentience in animals, we shouldn't be dismissal of the possibility of plants being able to pass that test, insofar as, if we think about it, what's happening when you are actually putting an animal to sleep with some anesthetics? Effectively, what is happening is that you are blocking, the action potential from happening, because you are messing up with the membrane properties, right? So ions cannot cross the membrane of the neurons. So that's effectively blocking, not allowing the action potential to fire. So the animal cell is not firing the action potential. And in the case of the plant, exactly the same thing is happening. The only difference, of course, is that we are speaking of different electrophysiological profile. So the wave of the depolarization, repolarization, and hyperpolarization, so the threefold wave, the profile is the same, but of course the scale is different, right? So in plants it takes much longer. The ion channels involved are different. We're speaking of calcium and not sodium, but those properties aside, the functional profile itself is the same. So it's no big news that when you have a plant and you see that it's sensitive to anesthetics, you can explain it in the same way that you would explain that it's being sensitive when it's an animal neuron, the one being anesthetized, right? The one being sensitive or being messed up its properties through the drug, right? Okay, so this would be a good way to move out of the animal kingdom and provide a metric space that is truly neutral with regard to kingdoms in the tree of life. Now, what happens in the case of associative learning or enhanced forms of associative learning? One of the basic examples of associative learning, we know from a study back in 2016, is that pea plants, the same plant that was anesthetized as well, pea plants, can be trained, in principle, at least that what it looks like, to condition the response to a stimulus that initially is neutral, right? So when you are exposing a pea plant to a fang, to an airflow provided or being triggered by a fang, you could see that no response is being conditioned. But when you pair the blue light, which of course the plants would be responding phototropically to the blue light, with the fang, you could see that after the pairing has been done, the response to the fang can be conditioned. So the fang would be triggering the tropic response, the directional response. So the plant would be growing towards the fang, expecting the blue light to come next, right? Now, this is when we get to my worry about how impatient we have become in the study of plant learning. This is a recent paper, 2021, by Ignacio Loy and colleagues, in which they were reviewing the literature on forms of learning in invertebrates, in plants, in Protista, and seeing how much work had been done and whether replication studies had been attempted and had yielded positive results, right? So surprisingly, what we find is that despite all this literature in which we repeat and repeat and repeat that some invertebrates or plants or Protista are able to perform this or that competency, we find that replication studies many times either have not been accomplished or have not been successful. In the case of associative learning, the study by Monica Galeano et al. In 2016, we have a study by Casey Markle, this one here, 2020, that was reporting negative evidence. They couldn't find associative learning in pea plants, right? And then we have all this literature in which back and forth they are responding to each other, like there is lack of evidence in the original study, then a response by the original team, a response from Casey Markle to Monica Galeano's team, etc., etc., etc. And what bothers me, and this is what I really wanted to discuss with you all, what bothers me in all this discussion is that we are missing something really basic that it worries me that nobody is speaking in this literature. So you have all these responses, all these replies, all these commentaries, one team responding to another, in which they say something is wrong, and then no, something you were not able to replicate properly, and they are missing something very obvious to the as long as you pay attention carefully. And here you have the Y maze, okay, the Y maze in which pea plants were growing, okay? So this is where your pea plant is growing, so it can grow towards the light, it can grow towards the fang. We want to do follow the protocol, the learning protocol, and see whether it will grow towards the fang when the blue light should come next, right? What's missing here, if you think about it, is that we are not able to see the plant. So we are trying, this is a behavioral study in which we cannot observe behavior. Do you realize how awkward this is? So we are not able to observe behavior in the first place. So this is an opaque Y maze. So all they were able to do was to look at the Y maze from the top at the end of the study, after the test day, and check whether the pea plant had grown into one arm or the other. But you just haven't got the slightest idea as to what's happening in between, right? So we need to peek into the Y maze to see what's happening there, right? So we did this recent study. We call it the 11 issues paper, in which we went through all the experimental flaws, the flaws in the design, in the experimental design, that nobody had paid attention to, despite, I insist, despite all this literature being published on this commentary and this response to commentary and this response to the response, and nobody was saying, hey, but look, we cannot see the pea plant growing one way or the other because the Y maze is opaque. Something as simple as that, right? So this has to do with how impatient we are, okay? Planta sapiens and human impatience. So once we go through all the problems that we should have dealt with in the first place, this is just a table. I won't go through all of them. I will just illustrate it with a few of them. You'll see that there are many, many, many issues that should have been controlled for prior to running the experiments or discussing the positive or negative results. This is just the way a pea plant grows, okay? This is how a pea plant grows if you crack open the Y maze and are able to peek through. So this is pretty slow, but you can see how it's growing, okay? So this is a plant as it grows, reaching the bifurcation point at which it needs to make a choice, either grow into this arm or grow into this other arm. So just pay attention to how important it is for the pea plant to be at the very center of the Y maze, because if it's not at the very center, being the bifurcation point, just this tiny point, just growing 1 inch to the left or 1 inch to the right would effectively make the pea plant grow straight into the arm. So no choice would be made after all. If you thought they were choosing to grow onto the left or to grow into the right, they might not choosing at all, but simply grow into one arm or the other if they are not placed at the very center. Of course, you need to be able to peek through to see how it is doing it. On the other hand, it might be simply bumping onto the bifurcation point. It might be growing in a straight line, bump onto the bifurcation point, and simply, out of mere growth, eventually slid into one arm or slid into the other arm, okay? So look what happens. Now this is a transparent Y maze, 3D printed, so we can time-lapse with infrared cameras, okay? And we can see the pea plant. Now you see the pea plant growing. Watch out, because this video is great to illustrate this point. See the pea plant growing into the Y maze. That's a transparent front, so we can see through the Y maze with infrared cams. So now the pea plant is growing at the center. So this is the bifurcation point. This is one arm, the other arm, and see, the tendril gets stuck onto the bifurcation point. And eventually, as the plant keeps pushing, because it keeps growing, eventually the pea plant will slide into one arm. You see, it gets off the hook because it was stuck at the bifurcation, and eventually, zoom, slides into it. You see, Now, if you simply looked at this plant after the last test day, you would report that this plant had grown, had chose to grow into the arm to the left. When we know, if we time lapse, that that's not happening by any means, that the plant is simply stuck at the center and eventually sliding, moving into one arm by mere physical force out of growth. You see? So imagine how fooled we can be if we are not careful in this type of studies. Now, another issue is that we know how sensitive plants are to blue light, okay? They are extremely sensitive to blue light. So you'd better make sure that you keep them apart in different compartments, because if they are not put apart in different compartments, the blue light from one cabinet will be influencing the behavior, the phototropic behavior of plants in a different Y maze. And in the original study from the videos, the supplementary materials, we can see that they were all on this tray where the blue light of one Y maze could affect the blue light or the behavior of the pea plants on a different Y maze. So imagine how important this is, right? At the same time, we even have an issue with the intensity of the LEDs. So all LEDs coming from the factory, when you buy a set of LEDs, they are not providing the same intensity. You need to control for that, because plants are going to be very sensitive to the amount of photons hitting on the tendril or the leaf surface, right? So we need to be extra careful here. You see, we have different compartments and they are enclosed. This is just open for you to see, but when this is all closed, they would be completely set apart. So there won't be any light leaking from one compartment to the next, right? But more importantly is in relation to this idea that I was telling you before. If the plant is not placed at the very center of the pot, the plant will be simply growing into one arm or growing into the other arm. And if your Y maze is opaque, when the plant comes out of the soil, you have no way of telling whether it's at the very center or not, because it was covered with soil, you see? So if it's covered with soil and you stick in the Y maze, then you don't know if it's growing out of the soil at the very center. In fact, it's impossible. Now, all the trials we did, they will always would grow 1 inch one way or another. So we need to use actually a laser liner to make sure that the plant is precisely growing at the very center. And then when you are transplanting the plant into the cabinet to time-lapse them, right? Once you put the Y maze, you need to make sure that the plant is actually, when it's coming out of the soil, it can be located at the cross of the laser liner, right? Once you do that, then you can put the Y maze on top and you are 100% positive that the plant is growing on the plumb line, such that it has a choice to be made. It has to decide whether it grows left or right and not simply grow into one arm because it was already growing into one arm from the very beginning, right? So this is all issues that appear like preliminary issues, but that are absolutely essential if we are going to have reach any solid conclusion one way or another. So this is the end result. You would have a recording cabinet that would look like this. Then you would have the possibility to peek through the transparent front of the Y maze within a closed place in which you see the Y maze is not stuck into the pot, but it has this lid. And having a lid is really important, because if you don't have this lid to close the pot, then you are encountering the following situation. When the pea plant is growing out of the pot, it could be facing left, it could be facing right, it could be facing anywhere, right? Because the tip is facing somewhere, right? So the way that the tip is facing is going to affect the pattern of growth, because it's nodding, the head is nodding. So that's going to affect the way it circumambulates. So we need to make sure that that is placed not just at the center of the pot, but symmetrically so that it's facing neither left nor right. So here you see a pea plant coming out of the soil, which is facing left. This one is facing right. You see how it's facing right here? And this one at the center is just looking straight. So this is the position we would want to have. We would want to have a pea plant that is growing out of the soil at the very center, provided by the laser laner, and in such a way that when you attach the lead, the Y maze with the lead, you can twist it, you can turn the lead such that as you turn it, it faces the camera straight front, right? You see? So that's what allows you to have a picture along these lines in the center in which you are ready to start time-lapsing, right? So I know this is all tiring issues, but bear with me because this is the last slide, then we can get to discuss all this, which is what I think it's important for PhD students and early postdocs. All these issues, I insist, are issues that you don't get to see when you read a paper. So if you read a paper on this, the paper that was reporting the alleged existence of associative learning in plants, you wouldn't see any of this. But they were attaching, for example, they were attaching the LED light and the fans to the side of the arm. So if you have the arm, they were attaching the LED to the side. So if the LED lights, the blue LED lights are attached to the side, the patterns of reflection are different in each Y maze. So we need to align them, the LED here, the LED at the center, to be centered in the very middle of the arm together with the fang. So now we have the fang and the LED at the very center, and that allows you to ensure that all plants are receiving the light and the airflow from exactly the same direction and the same angle. There is no difference in between them. But none of this was done either, right? So imagine if you are having all these issues that you need to deal with them prior to having any solid evidence, any evidence that we could call solid. Imagine how difficult it becomes to decide whether we have any conclusive evidence, because all of this ought to be taken into account. So we know that the fact that the more intense both stimuli, the condition and the unconditional stimuli are, we know that the more intense they are, the greater the effect of the training is going to be. But if we are not controlling the intensity of the blue light or the angle or the way it is impacting on the surface of the plant, we don't have a way to control whether more intensity equals greater effectiveness, right? In the same way, we would need to control the interstimulus interval, the amount of pairings, and like this, many other criteria that altogether point in the direction of a robust body of evidence, hinting at the possibility of truly associative learning, right? Okay. So now back to the indicators or markers that Jonathan Birch was mentioning. They were mentioning enhanced associative learning. So think about trace conditioning. When we are having trace conditioning being tested in animals, we might be thinking that trace conditioning was something that simply belonged to mammals, or we might think that it goes way down or to more distance field. So now we know that trace conditioning, we have this gap in between the CS and the US, as you have in trace conditioning as opposed to delay conditioning. We know now that type of enhanced associative learning is being exhibited by the fruit fly, or that is being exhibited by honeybees. And that's something we weren't thinking of a few years ago. Now, is it possible that pea plants are able to perform enhanced associative learning? Well, we really don't know, but if we are not implementing all these measures, if we are not having into consideration all these controls and all these pilots and all these careful studies that we need to do prior to running the main experiment, there is no way we can tell whether they are performing this or not. We might have an inclination, we have a hunch, we might decide to put our money to bet empirically that they won't be able to achieve it. But this is all wishful thinking, because we don't have the experimental means to solve the matter, right? So whether plants are able to learn by association and whether plants are able to exhibit more enhanced forms of associative learning is something that we cannot discuss theoretically, right? We can only solve the issue by running the experiments in this way. But unfortunately, this is something we are not doing because we are not being patient enough, right? So when in my book, In Planta Sapiens, when I discuss all these issues, there is a tendency, or it might be far too easy to place the burden of proof aside and claim or complain that the skeptics are not understanding what you mean to say, that the skeptics don't truly understand what we mean by plant intelligence or by plant sentience, or that they are obsessed with animals and they are falling prey or animal-centric biases, etc., etc. But I don't think that's a good lesson to extract from these debates. I think we should be doing this work. We should be way more self-critical than we are being. And being self-critical allows us to do way better than simply say, hey, they don't understand us. And this takes us to the issue of whether we are reporting quick results with way far too much risk of falling prey of confirmation biases or reporting false positives. When you look at all the issues we've identified, you realize that we need to slow down, to be way more careful, and maybe instead of publishing 10 papers in 5 years on the topic, to maybe just publish 1 paper in 10 years. I know this goes against the trend nowadays of fast science, but there is no shortcut to robust data gathering. There is no shortcut. The only way to go is to do slow science. And that means that despite the breaking news, the headlines that all these studies are making, they are making a lot of noise, but they then evaporate the next day because they are not solid. And that's a shame because we are distracting our students, our young researchers from following solid research lines by picking or by resonating with sexy or appealing headlines or appealing breaking news that end up not driving you or not taking you anywhere, right? And now let me say just a few words. I don't know how much time we have left, if it's been long enough, but let me say a few words on not on the types of behavior, of plant behavior that we want to unearth, but in terms of the psychological framework that we are trying to do this from, okay? So in comparative studies, sometimes, I know there is a tradition of behavioral studies, but there is another tradition of assuming a cognitivist understanding of psychology, as if all psychology was cognitive psychology, in which by default, any psychological competency that we are able to identify is meant to be inferential. So the environment is not rich enough. So the poverty of a stimulus makes us build or construct a framework in which somehow we have to cognitively solve the problem from the inside out, right? So it's an indirect, inferential, cognitive, algorithmic, cognitivist framework in which we are trying to find the algorithm, the software, the recipe that our brain mind is following to solve the problem or to approximate the input-output function, right? So to speak. We are summarizing 80 years of psychology in 2 sentences, but you see what I mean. Now, against this is great. There is no problem at all with cognitive psychology. In fact, I will defend the opposite view. But we need to understand that other frameworks might throw a lot of light upon comparative efforts. So when we are exploring through a comparative lens the behavioral patterns of the psychological doings of different species, we might end up realizing that a non-cognitivist ecological framework delivers a different set of goods. And that might be more informative and more enriching. So in this case, we would be speaking of proximal and not of inferential, direct and not indirect, behavioral and not cognitive, and picking on details of implementation as opposed details of the algorithmic recipe being followed, right? So I'm not claiming that we should be using exclusively ecological tools as those provided by ecological psychology schools of thought. In fact, what I'm claiming is that we should be approaching them both in a collective manner, in which we should ask or invite our students to team up in teams of adversarial collaboration with people coming from a cognitivist school, with those coming from an ecological school, with those from a more behaviorist inclination, because by approaching the issue from the theoretical commitments that all these schools of thought bring to the discussion, we have a way richer way out of the problems that we are facing. So it is not that we should be following a paradigm, but it should be multidisciplinary and multidisciplinary in terms of both the non-psychological tools that are being needed to be brought into the discussion, as well as the different schools within psychology itself. So this is just an example of what it would look like when we think of trying to understand plant behavior. In this case, take the example of the cuscuta. Remember the cuscuta reaching or choosing the tomato as opposed to the wheat plant. If you are thinking of an ecological, the way an animal exploits ecological information in the surroundings, so denying that the environment is poor, by moving around the subject, the agent, if I'm moving around in my surroundings, I'm creating an optic flow, say. So I'm creating an optic flow in my retina. So information is flowing in the form of this optic flow that as I'm approaching a target. So if I'm approaching this target, this image is expanding in my retina. If I'm moving away from the target, the image is shrinking in my retina. So you can apply these patterns systematically and try to look to search for the type of ecological loss that an agent might be following. So by moving, we are creating flows of information, and those flows of information are telling us, are informing us as to how to close the loop and to keep moving. So this closure of this loop of perception and action through ecological means is informing the subject, the agent, as to how to keep progressing. So if you think about it, this is not different from what happens in the case of the plant. We are simply changing, and this is critical, the idea of an animal locomoting, me locomoting towards the target, with respect to the plant, which is also locomoting towards the target. In this case, by locomoting, we mean growing. So a plant that is growing towards a target, say a cuscuta growing towards a host tree, is doing the very same thing I'm doing when I'm growing towards the target. So if we think of this image expanding in my retina, we can also think of energy flows that are being created as the plant grows with respect to the surroundings. So if you go back to the previous example, you can think of a plant growing and sensing or telling, being able to tell through ecological means and not through cognitive means, right? So ecologically speaking, could be tell the amount of neighbors in the vicinity, who is around, who are the potential hosts, for example, by sensing how the ratio of the red to far red light changes with respect to its pattern of growth. You see how we are closing that loop we said before? So as the plant is growing towards, so say it's growing left, if the ratio goes up or down, remember, if there are plants around, they will be sucking, absorbing red. If there are no plants around, they won't be absorbing red. So you can see the ratio of red to far red light, how informative that is of the amount of competency in the vicinity, or the amount of hosts, potential hosts for a cuscuta in the vicinity. So as I grow, I can see how the ratio drops or peaks with respect to my directional pattern of growth. And then ecologically, the plant can gather that information, resonate with that type of information, and decide to move or to keep moving But here in this case, I hope you can see that by decides, now I'm not speaking of decision making cognitively speaking. I'm thinking of decision making ecologically guided. So in terms of how information is guiding further patterns of growth as the plant keep growing. Oh, now the ratio is going up. Oh, now the ratio is going down if I twist and turn left and not right. Now think of the pea plant growing into the Y maze, how that applies equally, right? So all in all, the two messages I wanted to share with you, one is that we've got to be way more careful, and we've got to slow down, please. We've got to slow down in the way we are doing science. That's a message that we should all convey to our students, because we are living in a hectic scientific world in which the name of the game is the next breaking news, breaking news, breaking news. And you end up having a lot of anecdotal data and not a single robust line of research in many cases, unfortunately. The second message I wanted to convey is the idea, on top of doing slow and not fast science, of teaming up with those that are not trained in your field, those that are not trained in ecological psychology, if you are a Gibsonian ecological psychologist, or the other way around, train or team up with the cognitive psychologists if you're an ecological psychologist. So these teams of collaboration are way more enriching than we initially thought they might be, because by confronting all these alternative working hypotheses, not from within one paradigm, but across paradigms, we have a clearer picture of all the possibilities that open up in the landscape. Sorry if that was too much or too long, but I think that's it.
Thank you so much, Paco. So we have some time for questions. I don't know if somebody wants to open your microphone for some questions, or also you can write the question in the chat. So go ahead, Anne.
Yes, can you hear me?
Yes, perfectly.
Yes,
I can hear you,
okay.
but I cannot see you. I can only hear you.
Yeah, let me turn my thing on and get some light on here.
Thanks.
Yeah, I'm having some trouble with my audio, so I just want to check. But I just wanted to say, first of all, how much I appreciate that. And I also want to refer everybody, and you in particular, back, because I think that we are all familiar with a long-term attempt to separate humans from primates and then primates from other animals. And each of those fails because it's really a negative enterprise, as opposed to taking, you say ecological, I would insert evolutionary,
Certainly.
which simply says, what is the problem? How does it get solved? And what are the parallel solutions given where the organism comes from? It's almost, let's extend the concept of Umwelt to all organisms.
Sure.
And say they have a sensory world of some sort, and they solve problems. So I'm wondering if you can kind of, just inserting a few more things like terms like that.
Absolutely. So, I mean, a field of research carries the name phytosemiotics. So in biosemiotics, we speak of phytosemiotics. So there is meaning making being done by plants. Now, there is a particular, so there is something specific about our approach, and it's that now, if you are bringing into consideration evolutionary or developmental biological constraints, we are doing one thing. So initially, within the plant sciences, okay? Within the plant sciences, we sometimes have a resistance to understand plants from a reductionist perspective, of course. So we want to bring, on top of the cellular and molecular biology, or the plant electrophysiology, or the plant biochemistry, we want to bring the evolutionary and the developmental biology, right? We want to bring in the ecophysiology. So one thing is to bring the ecophysiology built in within this evolutionary and developmental framework. But on top of that, the twist is that what we are doing is bringing all that combined effort from the plant sciences within a cognitive science perspective. And that is what was missing until recently, that the best plant scientists were doing was to say, hey, we need to enlarge or enrichen or combine plant electrophysiology with ecophysiology or evolutionary biology. But the questions being raised were still being raised from within the plant sciences writ large. And that means they weren't genuinely tackling the issue as to whether plants make decisions. For example, are we speaking of choices metaphorically or we mean it literally? No, we do mean it literally. But to understand why, we need to move out of the plant sciences and have a cognitive science perspective on any form of life whatsoever in the tree of life. So I agree with you that we need to have this more embracing, encompassing perspective that applies to the whole tree of life. But to do that, we need to use as the entry point the cognitive sciences as such. Now, within the cognitive sciences, then we have this other discussion as to whether the framework is cognitivist, constructivist, ecological, pragmatist, whatever, whichever school of psychology you are thinking of. But first things first, it's got to be trained, coached. The questions have to be coached from within the cognitive sciences, right? Because otherwise, we would be speaking of adaptive behavior. Even if you bring the phylogenetic or the evolutionary perspective, it could be still a question of adaptive behavior. And we are not speaking of adaptive behavior. We are speaking of which types of behavior that are adaptive. Of course, they've got to be adaptive, otherwise we wouldn't be speaking of cognitive sciences, are or deserve what sort of behavioral repertoire, adaptive behavioral repertoire, deserves the label of cognitive or intelligent. That's when, okay, all these considerations about flexibility, about degree of anticipation in the behavioral repertoire connect to meaning making. Or it's meaning making in sensing those ratios. And it's not simply responding, reacting to the ratios of red to far red. No, it's integrating that information with a lot of other information in order to choose, to decide. And now we are not anthropomorphizing it. The plant is deciding in terms of the meaning making it's doing of its surroundings to grow left towards the tomato as opposed to right towards the wheat. So, but I certainly would agree with you that that's very much needed to bring in considerations that relate to biosemiotics. Thanks.
Thank you so much. And Jose Burgos, go ahead.
Thank you. Great talk, Paco.
Thanks.
Wonderful. Very thought-provoking, very inspiring, and very humbling. It only confirms my anxiety about how much there remains to do.
Absolutely.
Everywhere. I mean, and I'm all for patience, of course, but everywhere. I mean, patience is good. I mean, not just in psychology, but also in physics and in everyday life.
We all have to chill out.
Yes, right. Exactly. But I understand. I understand that there are a lot of very, very urgent problems that need solving, and society, imposes this, or requires fast results, because much of this research is being paid by the taxpayers' contribution. So when someone asks me, what are you doing with my money? And I start this long, convoluted tale about, the apparatus and the method and the procedure, and they start, yawning at best. So I'm all for that, of course. And I'm all for slow science, although slow might not be the most flattering qualifier. Probably careful science.
Sure.
Careful science, and we must be careful. And I'm all for this. Now, I don't want to monopolize the conversation, but thinking about definitions, which is a major topic here. I always wonder what scientists in general, psychologists included, seek with their definitions. Right? And it's far from clear to me exactly what definitions do in science in general. I mean, my question is something like, what's in a definition? And I have found it very, very useful and very liberating, in fact, to look at this in terms of the very old, the ancient distinction between nominal definitions on the one hand and real definitions on the other hand, where nominal definitions are just conventional stipulations for the purpose of abbreviating, where you use a certain term as a shorthand, as an abbreviation for another longer expression that you don't have to repeat. If you look at it that way, and you use those terms or define those terms nominally as mere abbreviations, I see no problem whatsoever. If you say, well, for the purpose of this research or this paper or this book, by behavior, and I quote unquote, by behavior, I mean this and this and this, quote unquote, and that's it. Without intending it to have any metaphysical, ontological, import about the real, true nature of behavior. Because as soon as we start thinking about it in those terms, as a search for the true nature of whatever, behavior, intelligence, consciousness, we get into trouble. And troubles really begin in really forcefully. So if you're defining nominally all of this, consciousness, intelligence, I have no problem with that. But when someone, and I could do it myself, I'd rather not. I'd rather take the eliminative path, which is just I don't use those terms. Period. If they cause so many problems, these terms like intelligence, behavior, I know it's hard to do to stop using this, but it's a path, it's an alternative, right? But if someone, and this is the end of my intervention, if someone tells me, well, if I'm defining nominally something, consciousness, for example, quote unquote, and someone tells me, but that's not what consciousness really is, my reply is, you're missing the point, because I'm not giving you a real definition of consciousness. I'm giving you a real definition would be a statement about the real, true nature of something. I'm not giving a real definition of consciousness. I'm just using the term consciousness as an abbreviation for something longer. Thank you.
No. Thank you. I entirely agree with you. I mean, I can hear you. Also about the money from taxpayers, right? How sensitive we have to be to what we do with their money.
Exactly.
But no, getting to the heart of your point, I entirely agree with the proviso that actually that's something I always tell my students. I say, well, if you feel uneasy with this, if I say plant intelligence or whatever, if you are not quite, something funny, you are uneasy with these terms, is not gonna let you sleep tight. Call it. I always tell them, like half joking, call it chumpituki. No, llámalo chumpituki. If you don't like it, call it chumpituki. I don't care what label you use, because to me, the best way to see that it wasn't that important in the first place is that as soon as they remove the label, they open up their mind to explore. And that's very revealing as to what was actually at stake. So to me, the one and only problem, the one and only danger of definitions is to think that you are signing a blank check, that they've got to be set in stone, come rain or shine. But they need not be set in stone for experimental program to get running. So you can get the ball rolling with provisional definitions, with tentative definitions. And in fact, sometimes the less set in stone they are, the more provisional or the more tentative, the better they are in initial exploration phases. Because we put too much stress in the actual major experiment we are running, as if the pilots of the experimental or the exploratory phase was not that important. And to me, the truly important part is the exploratory phase. Once things are not so constrained and allow you to explore wider landscapes, terrains, right? So to me, what's really worrying about the use of labels or definitions is when somebody, as well, you know, from the exchanges in Animal Sentience with our target article. So for those of you who don't know, we published this target article in Animal Sentience in the journal on plant sentience, and we received a number of commentaries from different authors. So Jose Burgos among them, right? So one of the authors responded like plant scientist skeptics from orthodox plant science. They responded something like, I mentioned about neurotransmitters, like saying, oh no, plants cannot have signaling molecules, the ones we speak of as neurotransmitters, GABA, glutamate, because there are no synapses. So if you think how awkward this line of reasoning is, wouldn't it be easier to simply say, oh, provided that we have these molecules playing this signaling role, let's revise our definition of neurotransmitter. Maybe it doesn't require a synapsis to be breached. So it's easier to keep a definition as a work in progress as long as it can incorporate all these breakthroughs, all these heterodox approaches, however preliminary or provisional they are. So I entirely agree with you in that, so far as you call that.
Yeah, conceptual flexibility is key.
Absolutely.
Thank you.
Thank you.
Thank you so much, Jose. So Mauricio, please.
Hi. Great talk. I read your book with a lot of
thanks.
So you can tell.
You did your homework.
Yeah. And I love your talk even more than your book, because in the spirit of parsimony and in the spirit of Morgan's canon, that we comparative psychologists value a lot, that your attention to detail of procedure, and that really buys me. I wanted to do what you ask everybody to do, which is to bring you out of the comfort zone. So I want to ask you a question that brings you out of the plant.
Okay, good.
And into the animal world, because I'm interested in the evolution of animal learning.
Okay.
And what I want you to speculate, because this I don't think, well, I think it's mostly speculation. It's on the fact that it is a fact that animals have evolved neurons, right? But it is also a fact that other living organisms that don't have neural components have a number of problems to deal with, like sensing stimuli from the environment, and like you said, making decisions, like you said with the quote, and so on. So all these things are obviously true, but it's also obviously true that we have neurons, that animals have evolved neurons. We have neurons, the sense that they have evolved, right? So tell us what you think about what neurons are contributing to learning, to plasticity, to cognition, if you want, or having to do with that you were trying to prove.
Sure. Yeah, definitely. No, thanks. Great question. Yeah, no, definitely. There is a clear divergence here, and it simply has to do with whether you've got to be fast to respond or not. So plants, in fact, some people argue along the following lines, that you've seen that in Planta Sapiens. Some authors say, hey, plants, they can afford to be stupid, because being sessile, being rooted, they can afford to be stupid. But animals that have to be fast, running, hiding, hunting, prey, we cannot afford to be stupid, and we have evolved this neural tissue that allows you to be fast, right? Okay, plants and animals are neither fast nor slow. They are tuned to the time scale they need to be in sync with. So plants are not stupid because they are sessile or because they are slow. They are simply not slow. They are as fast as they need to be, provided what they need to do. Why? Because their strategy, evolutionarily speaking, is a divide and conquer strategy. So of course I need to be fast, and to that end, neural tissue comes handy, because I need to be fast because I have one single heart. If you pick my heart, I'm done. So I better be fast to hide in front of a predator. But in a plant that is sessile, because the plant cannot possibly flee, the plant has evolved a different set of adaptations. In this case, not to have any living organ located in one particular spot. So everything is decentralized, fully distributed. So you chop me a branch, I grow another branch. I don't care. I don't have 2 kidneys, 2 lungs, right? So that's one of the reasons there is that difference. But I insist, it is still very anthropocentric to measure or to gauge the degree to which they are fast or slow as opposed to ours. They are not slower than we are. They are as fast as they need to be, provided the type of problems that they encounter. And one of the problems that we are encountering different from theirs is that, put it the other way around, plants could say, hey, you've got to be damn stupid if you need to go shopping to cook your dinner. I do photosynthesis. No, it's neither worse nor better. It's simply different problems, different solutions. So we are encountering different problems, we are putting on the table different solutions. In our case, certainly neural tissue comes handy if you need to provide fast responses, because we are not decentralized to the extent that they are.
Yeah, great. Yeah, I agree. I agree. There is something to do with movement. There is something to do with also animal size. That animals, there are a few phyla of animals that don't have nervous systems, and they tend to be rather small. And
sure.
if you are going to evolve large body sizes, you need fast communication also. So that
but sequoia trees are huge.
Yeah, I mean, they have a different. Yeah, it's like you said. Yeah,
so comparative studies. We cannot fall prey of this skeleton toy understanding of the comparison. That's what I meant to say. So this toy understanding of what is it that we are comparing doesn't allow us to zoom back and enlarge the picture functionally. So this is a functional comparison. So they are doing what they need to be doing by other means, but bioelectricity is the name of the game in nature. So plants are actually, they evolve action potentials. So plants fire action potentials, not just the Venus flytrap or the mimosa, any plant whatsoever. So why good plants need to fire action potentials if they are not animals and have no neurons? Because action potentials is not tied unidirectionally to neural tissue. So action potentials pays off in non-animal forms of life.
Yes,
we need to have this more abstract functional.
I agree. I don't see much of a problem, because we know that evolution is a process that's very opportunistic, and so I don't see, if you have a set of if you have molecules that react to light and you don't have many of them, you're going to use them over and over again in a variety of roles, a variety of functions. So rhodopsin is present in animals that don't have eyes, that react to light, or in plants. So you have this concept of convergent evolution, right? They have that having similar ecological problems will provide for very similar solutions. They may not be the same. They may not be the same. And this is the danger of the danger of equating behavioral data, including growth, in animals, plants, or whatever organisms that are very different, that have evolved independently. The danger of equating similarity in behavior with similarity of mechanism is the problem. So we have to recognize that you can achieve similar solutions to problems, or not you, but that living organisms can achieve similar solutions to problems, to environmental problems, through different means.
Oh, certainly. I mean, there is.
But to me anyway, the interesting part is what are the common aspects, because there might be many ways to store information, for example, to have plasticity, behavioral plasticity. There might not be many ways to do that. And so there might be, let's say, ion channels, or there might be certain things, like you pointed out, to neurotransmitters. There might be certain things that are present all over the place. They have been used and reused in different ways in evolutionary time to solve similar problems. And so there might be a level of a common ground level where where nature simply doesn't have enough or infinite numbers of ways of producing something. For example, there are not many ways to do synaptic plasticity. So you have cyclic AMP and Krebs, and these processes are common to animals that are otherwise very different and have evolved independently for hundreds of millions of years. And yet the processes are the same at the cellular level, are very similar. So there might be something, some sort of drastic constraint. And I'd like to understand that. For example, what's common if you have plasticity in plants or fungi or bacteria or protists? If you have plasticity there, what is it that is common between them and animals? Because there are at the macro level, there are substantial differences, but there must be some common ground underneath that allows for those capacities.
Well, that's interesting that you brought bacteria, because you see, to me it's really intriguing that many researchers find it way easier to discuss decision making in bacteria than in plants, right? And why is that the case? Because in bacteria you appreciate locomotion, swimming up a sugar gradient. So as bacteria swim, you are able to empathize, to put yourself in their shoes in a different way. So our problem is that we are biased towards locomotion. And even in unicellulars that locomote, we are able to bridge the gap in between them and us. And that's, to me, really awkward that we have such a, blind eye. We turn a blind eye to any pattern of behavior which is not based on locomotion. If you think, Jose Prados in the chat, he was saying, a neuron is a cell after all. Of course, you can find action potentials all around, right? Certainly. But you know what the problem is? That because we turn the blind eye to behavior based on growth and not locomotion, in the case of plants, action potentials are approached as something anecdotal, something that happens in the Fast and Furious, in Venus flytraps, in mimosa. So those plants that you can observe to the naked eye. So if I can observe them to the naked eye, it's easier for me to put myself in their shoes because they are kind of closer to what I mean by locomotion. Then I grant them those action potentials and the role they play. But that allows us or prevents us from truly unearthing what's universal, and is that action potentials rule electric signals are part of what explains systemic signaling in plants, in any plant whatsoever, not just in those that we can appreciate their behavior to the naked eye. So that is, to me, the problem, that if you're able to appreciate this in unicellulars because they locomote, and then you turn the blind eye to those features allegedly being shared across the tree of life, once they are being exhibited by a system that is sessile, then the problem is not the mechanism or the details of the implementation. The problem is that we don't resonate with those types of behavior that, look like more distant to what we mean by canonical or the gold standard behavior. And that is what we cannot get rid of our understanding of behavior as the gold standard. That's what is preventing us from putting them on or understanding that we are all on the same page. And to me, it's paradoxical that we are able to appreciate it in the case of unicellulars and not in the case of multicellulars that don't work. Yeah, but thanks.
Thank you so much, Paco. We have some greetings here at the chat so you can read. So I think that was our last question. So again, thank you for being here with us. Thank you for sharing your knowledge with us. For those that are here, I encourage you to check our social media in Facebook, on Twitter, now X, and also on our YouTube channel where you can check our previous lectures from the society. So keep with us. We have a next talk this Friday, so we can close from the lecture series of this year. So again, Paco, thank you so much for being here with us.
Yeah, well, thank you, and thank you all.
So everybody have a good day. Bye.
Cheers. Bye.
Gracias, Paco.
Hasta luego.